Good morning, everybody. As Nisha said, I'm Heather Williams. I'm going to be introducing Dr. Stephanie Ware. Dr. Stephanie Ware is a geneticist. She is out of Indiana University's School of Medicine. She has been working on things related to heterotaxy for about 10 years. She does research and sees patients, so that's quite an accomplishment. And then she works a lot with all sorts of patients that have congenital cardiac conditions, and the research into that and trying to find a cause of why our kids develop these congenital conditions so that perhaps we have a chance one day of preventing those from happening. So Dr. Ware is extremely excited to be here. She is very willing to talk to families individually after her talk. So if there is something that you would like to address with her specifically, please make sure that you, her time with us is very limited, but do make sure that you speak with her. So at this time, I'm going to turn the podium over to Dr. Ware. Please join me in welcoming her to our first Heterotaxy conference.
Well, thanks very much for inviting me. I'm happy to see everyone here. I'm going to talk a little bit about genetic approaches to heterotaxy. I think the two talks after mine will probably be dealing more with some of the management aspects, but I'm going to talk about some of the research that we're doing to try to understand causes, and then from a clinical standpoint, really, what is the role of a geneticist or genetic evaluation in kids that have heterotaxy. So the first thing I'll do is just give a little bit of an overview of heterotaxy and how it relates to congenital heart disease. I think all of you will be familiar with this, but one of the things about heterotaxy is it is so variable, so you may not realize the full scope. And then talk, as I said, about the genetic evaluation of heterotaxy, the progress that we've made so far and the directions that we're going, and then end with talking about the research. And this is, some of it's very basic research, but as Heather said, the goal here is to get at what are the different causes, with an idea that if we can identify causes, then we can begin to try to address therapy and cures. So as all of you know, heterotaxy is really a multiple congenital anomaly condition. It's characterized by cardiovascular malformation, congenital heart disease, as well as abnormal positioning of other organs of the body. And this results from things that happen during early development of the embryo or the fetus. And we'll talk a little bit about some of the developmental biology behind that. So this is a really dense slide, not meant to read, but meant to show exactly how complicated heterotaxy is. So on the left side, you can see just a list of all of the different types of cardiovascular malformations that you can see in individuals with heterotaxy. And then on the right-hand side, it gives the distribution of heterotaxy or laterality defects. And what you can see there is that in terms of simple heart defects, the percent is relatively low, only about 7.4%. So the vast majority of individuals with heterotaxy do have complex cardiovascular malformations. So again, the point of this is that there's a lot of different heart defects that you can see in heterotaxy. It's not a single condition, and they tend to be very complex. And this has been, in addition to the clinical challenges that go with managing this because of the complexity of the malformations, it's been really challenging from a national standpoint in terms of doing studies on kids with heterotaxy, because heterotaxy isn't recognized as a single cardiovascular malformation or a single group of disorders. There's so much variability in it. And so for a long time, it didn't even have its own diagnosis code. So if you wanted to try to study or look at all of the kids and adults with heterotaxy, there was no way to kind of pull that information. So it's been very challenging. And then the other thing is, because there are so many different combinations of variability of the anatomy, it's hard to get really precise information if you want to try to understand in detail, well, what causes this particular form of heterotaxy versus that. So it's one of the challenges in the field, I think, and something that parent groups can do a good job of advocating in terms of record keeping and trying to keep the details of the different diagnoses. So heterotaxy is thought to make up about 3% of all types of congenital heart defect. That's probably an underestimate. We think of, from a genetic standpoint, we think of kind of classic heterotaxy, where you have all of the situs abnormalities as one end of the spectrum, and there's probably very mild forms of heterotaxy that don't ever get labeled as heterotaxy. For example, a child who didn't really have a cardiovascular malformation, but maybe had bilateral superior vena cava and had mispositioning of the liver and the spleen. To me, that's heterotaxy, but that might not get called as heterotaxy by the physicians that we're seeing that individual. So I think the 3% is probably an underestimate. As I said, there are abnormalities of abdominal organs in classic heterotaxy. And then something that's not really realized is that about 30 to 40% of individuals with heterotaxy will have some kind of midline defect. And most commonly that's a vertebral defect, something in the spine. Sometimes it could be a rib abnormality. And more rarely you could have problems in terms of the central nervous system. If you look at the epidemiologic data, it will tell you that there's an overrepresentation of males with heterotaxy. Some of the newer numbers aren't bearing this out, so I think it's something that's still under study a bit. But if you look at individuals with the polysplenia form, really equal representation, males and females, but the old data at least will say that with asplenia, it's more common in males than in females. So again, this is just a list. Those of you who've seen a lot of different doctors may get frustrated with the different terms that can be used for heterotaxy. So I tend to use either just heterotaxy or laterality disorder as kind of a broad category. But in the past, people tried to give it a lot of different names: right atrial isomerism, left atrial isomerism, asplenia, polysplenia, Eichmark syndrome, Cartageners. So there's a lot of different names that have been applied to it. And in terms of thinking about it as a physician, we try to group things together that make sense because it helps us figure out how to manage individuals. And so the polysplenia or left isomerism and asplenia right isomerism lists were generated with the idea that this would help people think about the patients. But I guess my experience at least has been that the individuals don't fall neatly into these categories. Sometimes they mix different types. So I find it easier to just describe the different anatomy, describe the specific cardiac defect, describe the abdominal organs, describe the spleen, and that makes it more specific to the individual with heterotaxy. There is an importance, though, that in terms of the right isomerism versus left isomerism, there's slightly different outcomes in those groups, and that is really related primarily to the fact that the severity of the heart defect. So asplenia type tend to have more severe heart defects or have a higher percent of heart defects. The one exception with polysplenia is that you're more likely to see biliary atresia in that classification, and biliary atresia is one significant cause of morbidity outside of the heart in individuals with heterotaxy. So in terms of the extracardiac management, when we see a newborn with heterotaxy, the things that should be thought about and should be done is all related to the anatomy and making sure that everyone knows everything that's going on. So abdominal and renal ultrasounds, vertebral x-rays, these are usually, don't have to be dedicated. If it's a baby, they usually will see that on the other x-rays that they're doing. Evaluation of spleen function, I think Kim Risma is going to talk about that in the third talk today. But the spleen is important for immune function, and the spleen, you can have a single spleen that doesn't function well with heterotaxy. You can have polysplenia, so multiple spleens that don't function well with heterotaxy, or you can have no spleen, which obviously is not functioning. And you need to know that because there's specific medical management recommendations related to that. Malrotation of the gut is something that can happen, and we always are vigilant to look for that in individuals with heterotaxy. There's pretty much a debate in the field if you know that the intestines are not are not normally coiled, whether they should be fixed or not, sort of prophylactically. And there's evidence both for and against that. It's the surgical literature is still divided on what the right treatment is for that. Hepatobiliary imaging or evaluation is important if there's a concern for biliary atresia, and imaging of the central nervous system if there's any concern there. It's not something that we routinely recommend, but it's something that we think about in every individual with heterotaxy. And then we'll talk a little bit more today about pulmonary and primary ciliary dyskinesia and the overlap between heterotaxy and primary ciliary dyskinesia. If a child or an adult has PCD, there's additional management recommendations related to that. It's not something that we routinely evaluate in a newborn with heterotaxy, but it's something that we think about because it impacts the long-term outcome. It impacts the child if they are on a ventilator before or after surgery, and there's specific pulmonary things that can be done to improve their function. And it's something that I think the parents of children with heterotaxy may have heard about or tuned into much more than the physicians. There's still only about 2 publications in the literature related to this. It's very new findings. So physicians are often unaware that there's this overlap between heterotaxy and PCD. So congenital heart, I'll talk a little bit more just about congenital heart defects in general and the genetics of them. So congenital heart defects are the most common birth defect. We see them in almost 1% of all newborns. And the morbidity related to heart defects hasn't changed that significantly over the years, although the surgical advances have been significant and children are being kept alive much longer now. The overall incidence remains steady. And with regard to the genetics of congenital heart disease, every medical condition is different in terms of how strong the impact of genetics is. So you have environmental modifiers, you have genetic modifiers, and the combination of that really determines the approach and the research, I think, related to those. So what you can see here is just some different disorders and the degree to which it's thought that environment impacts versus genetics. And you can see that for congenital heart defects, genetics plays a strong role. And the way that we get at this usually is from epidemiologic studies. So you look at national studies and you look at all of the children that are born with heart defects, and then you ask how often do other people in the family have a heart defect, and that gives you a clue that there's a strong genetic contribution. So this was a really nice study that was done out of Denmark. They have a national health system, and they basically for over a 40-year period collected information on birth defects on everyone that was born in the country, and then they went and looked at the family members, how many additional family members had heart defects, how many individuals who had heart defects had children with heart defects. So they compiled all of that information, and when they did that, what you see here is that at the very top of the list of heart defects is heterotaxy. So heterotaxy, of all of the types of heart defects that you can have, actually has the strongest genetic contribution to it. And this is despite the fact that in most families, if you take family histories, most people who have someone with heterotaxy in their family, there's no one else with heterotaxy in their family. So that's a little bit puzzling to people sometimes. Well, no one else in my family has it, so how can there be a strong genetic contribution? I think it's because the genetics are so complicated related to heterotaxy, and we'll talk about that a little bit more. So heart development, why is the heart so often affected in heterotaxy? Part of the reason for that is just that heart development is so complicated. So this is a cartoon just showing really how the heart forms. When the baby's first developing in the womb, you just have a group of cells that sort of look like a boomerang that are cells that are going to become the heart. And then from there, those cells become a tube, and it's a straight tube that's kind of in the middle of the developing embryo's body. And then that straight tube has to undergo folding. They call it a C loop and then a second S loop to actually get the ventricles and the atria in position with each other. So it's a really complicated process. It's kind of surprising that it doesn't go wrong more often than 1% of the time, in order to establish the really complicated two ventricles, two atria, and then the outflow tract. And everything has to be aligned properly, otherwise the blood doesn't flow correctly. So the next slide is going to show you, it's actually a video of heart development. It's by time-lapse photography. It's actually of a chicken embryo, but it looks very similar to what human heart development looks like. And I think you can see all of the twisting and turning that goes on. So this isn't the final heart, but that's the initial twists and turns that the heart has to take. And so I think you can get an appreciation for the fact that has to be very carefully coordinated. And it's the really genes that drive that. The genes are the information that tell the body how to form. And genes are telling the left side of the body that it's the left and the right side of the body that it's the right, and setting up really that blueprint. And so if the blueprint doesn't get set up correctly, and the left side doesn't know that it's the left, the right side doesn't know that it's the right, then this complicated twisting formation of the heart isn't going to happen properly. So I think that's one of the reasons that heart defects are so common in heterotaxy, is that the heart, starting as that linear tube and then having to undergo this looping process, the heart seems to be particularly sensitive to the confusion about left side versus right side. So with regard to the genetics of heterotaxy, pretty much any type of inheritance pattern can be seen in heterotaxy. So we talk about autosomal dominant inheritance, where you see something running through every generation of a family. X-linked inheritance, where males are primarily affected and females are kind of silent carriers of the condition. Autosomal recessive, where each parent is a carrier, but they're not affected. But then if the children inherit two abnormal copies, that the child can be affected. So we've seen all of those types of inheritance patterns in individuals with heterotaxy, but far and away the most common thing that we see is what we would call sporadic heterotaxy, meaning the child has heterotaxy, nobody else in the family has it. And our thought there, as I said, is that heterotaxy is very heterogeneous from a genetic standpoint. And when I talk to families, a lot of times what I'll say is it's kind of the gumball analogy, that you have a gumball machine and you put your quarter in and turn it, and the combination of colors of gumballs you get out is just dependent on the turn. And it's sort of the same with genetics. We know that there's a lot of genes that impact this development of the heart, and many of them would be too severe to actually be survivable if that gene were severely affected. But if you mildly affect one gene, mildly affect another gene, mildly affect a third gene, the right combination of those genes that are all mildly affected can result in heterotaxy. So that's one of the big challenges for us, is to try to identify what are the groups of genes as opposed to just a single gene. What are the groups, and what combination do they have to be in order for an individual to have heterotaxy? So rather than thinking of a gene as causing heterotaxy, that's more a model where we think about having a risk factor, thinking of having a susceptibility. So on the clinical side as a geneticist, what do I think about when I'm asked to see a baby with heterotaxy? I kind of think in four broad categories. So we'll talk a little bit about the ciliopathies and primary ciliary dyskinesia, and then I'll show you from the developmental standpoint why cilia are important and why they cause heterotaxy. Talk about syndromic heterotaxy, and then isolated heterotaxy. So my guess is that most of you here who have children affected have children that are affected with isolated heterotaxy, but we'll talk about these others as well. So we'll start just with some basic genetics. So as I said, the genes really are the instructions to our body in terms of how to form. They determine our hair color, our height, but also how the organs form. And genes are organized, are packaged into chromosomes, and we have 23 pairs of chromosomes. The last are the sex chromosomes, so paired in females and XY in males. And some genetic conditions arise because you have abnormalities of chromosome number. So Down syndrome is probably the most common genetic disorder that people have heard of. And Down syndrome was caused by, instead of having 2 chromosome 21s, you have 3 chromosome 21. But then if we look at a more detailed level, so if you zoom in on the individual chromosome, what you see is something that looks like this, and this is the chromosome blown up, and on the chromosome there's the individual genes. And then if you zoom in on the individual genes, you get down to the level of a DNA strand. So you can have genetic disorders that are caused by small deletions or duplications, which we call CNVs. You can also have genetic disorders that are caused by changes in individual base pairs. So there's a lot of different levels at which you can have genetic disease occurring. So this is what chromosomes look like. You have chromosomes in every cell of your body. And if you take some blood and you get the chromosomes from it, and they do a special stain on them that makes them look kind of like striped patterns, zebras, and individuals who are trained in this can look at these under a microscope, and based on the particular stripe pattern, they can recognize what chromosome it is. And they're pretty much numbered from biggest to smallest, so chromosome 1 is the biggest chromosome. As I said, if you've got abnormalities in chromosome number, that gives you a genetic syndrome typically. So often the way that I'll talk to families about it is like if you walk into a library and you look at an encyclopedia of books on a shelf, and if you do a chromosome study, what you're looking at is the number of books on the shelf, and there should be 46 books, and chromosomes count and see that there's 46. And then you can tell a little bit about the size of the books and the thickness of the books. So you know there's supposed to be a certain height, there's supposed to be a certain thickness. That's sort of the level of resolution of a chromosome analysis. So you can tell, as I said, with Down syndrome, you can tell that there's an extra book. Another genetic syndrome where you're missing part of chromosome 5, you can see that part of that book is missing. So that's what a chromosome analysis will tell you. So about 8 or 9 years ago, a new technique was developed called chromosome microarray analysis, or CMA, which allows us to look at the chromosomes in a more sophisticated way. So this, with the encyclopedia analogy, not only are you able to count the number of books and see that there's 46 books, but you're actually able to look at every page on the book, and you can tell, are there any pages that are extra, or are there any pages that are missing? And in some cases, having an extra page doesn't cause any disease, but in other cases, having an extra page does cause disease. And as I said, this testing has been available for about 8 or 9 years now, and we know some very well-characterized genetic conditions that are caused by extra or missing pages. We know some things that don't cause disease. They're just sort of normal variants in the population. And then there's sometimes still findings that we'll see an extra or missing page that we haven't seen before, and we don't know whether it causes disease or not. So that, because the test is still somewhat new, we occasionally will have things that we don't know exactly how to interpret. So this is just another picture of the same thing, showing that you can have basically pages missing or pages extra, but when you look at it in the context of the chromosome, it looks like this. If you've got here missing page C, then that shortens the chromosome, or an extra page C, that lengthens the chromosome, but you can't really see that on that chromosome analysis. You have to do the more detailed chromosome microarray analysis. So what do we know about these extra or missing pieces, or CNVs, as I'll use that term, and heart defects? So we know that about somewhere between 3 and 25%, and that's a pretty broad range, of patients with congenital heart disease and some other organ system that's affected will have an abnormal chromosome microarray test. And about 3 to 10% of patients with just isolated heart defects will have an abnormal microarray. We did a study several years ago where we looked at specifically at patients with heterotaxy and looked at the CNVs in those patients. So we had 262 patients with heterotaxy, and we found about 15% that had CNVs. Not all of them could we say were definitely the cause of the disease, but they weren't something that was seen in the normal routine population. And then another study that's come out more recently, 74 patients with classic heterotaxy from New York State, and they found 20 rare CMVs in 19 patients, so about 26%. So this is some of the newer information that's out, and it's one of the reasons now why clinically we say that any baby with heterotaxy really should have a chromosome microarray analysis done. So then the last level in terms of how genetic disease can happen is at that most detailed level, at the base pair level. And so this is kind of analogous to doing a spell check. So when you do the chromosome microarray analysis, you can tell if there's pages extra or pages missing, but you're not looking at the individual words on a page. And that's another way that you can get genetic disease, is if you have a misspelling on one of those words on the page, that can also cause disease. And we're going to talk about X-linked heterotaxy, and that's the way that X-linked heterotaxy happens. It's a misspelling in a specific word. So the specific type of analysis for this, it has to be done using a different technique. It can still be done on blood, but it's a different technique to look at those individual base pairs and do the spell check. And then, there's still one more level beyond that, which we're not routinely testing now, but even when you do the spell check, you could spell check every gene that we have, and you're not going to find all causes of genetic disease, because there's still causes that I would say are kind of the grammar or the spacing or the punctuation, and we don't routinely test that for that right now on a clinical basis, because we don't know how to interpret a lot of those results. But it's just showing, 10 years from now, this talk will be a little bit different, and we'll be talking about looking at the spacing, looking at the commas and periods as well. So, as I said, heterotaxy is primarily a genetic disease, but we're not very good at identifying the causes. We've gotten a lot better over the last 10 years, but we have a long way to go. We do know some of the specific individual genes that cause heterotaxy, and they're listed here. But there's, I think, an enormous potential still for discovering new genes that cause heterotaxy. And then, as I said, the ultimate goal, or the next goal from that, is to find out the combination of genes, with the idea that in a lot of cases it's not just a single gene that's causing it, but it's that combination. So, so that's kind of the background for syndromic heterotaxy as well as isolated heterotaxy. And then I'll talk a little bit about this overlap between some of the ciliopathies and heterotaxy. So again, it's relatively new. In the last 5 to 10 years, there's this whole group of disorders that we call ciliopathies. Cilia are just little organelles that sit on a cell. They can sense movement, and they were thought to be not very important until about 10 years ago, when we started finding out that they cause a lot of disease. So there's a whole group of disorders. They can affect the kidney, they can affect the development of the brain, they can affect eye, actually can affect both growth as well as limb patterning. Some sensory things, and obesity, and then so you'll see lateralization defects or heterotaxy spectrum disorders as well, and then affect the pancreas. So those symptoms fit into a number of names of disorders that I'm just going to kind of show up here. They're all really long, complicated names. I think the point of this is that it's a pretty big group of disorders, and heterotaxy is seen in only a minority of individuals with these, but it's something that that you need to think about if you see a child with heterotaxy, the fact that there can be a lot of other syndromes associated with it. So, for example, Bardet-Biedl syndrome, down here, only about 5% of kids with Bardet-Biedl syndrome will have heterotaxy. And if you look at well, of kids with heterotaxy, how many of them have Bardet-Biedl? Probably only 1% or so. So it's not common, but it's something that should be thought about, especially if the individual seems to be a little bit unusual. They're not sort of the typical heterotaxy patient. Should think about some of these other conditions. So then primary ciliary dyskinesia is slightly different than that other group of ciliopathies. And primary ciliary dyskinesia has been recognized as a disease for a long time. It's also called emodal cilia syndrome. So again, the cilia are just these things, they're shown here kind of in close-up, that line a lot of the body passages. So they're present in the trachea. They help brush things forward to keep dirt and so forth out. And if the cilia aren't working, if they don't move, they can't perform that brushing action, then there's susceptibility to lung infections, susceptibility to sinus infections, recurrent ear infections. And so it's been known for a long time that that individuals who have primary ciliary dyskinesia can have situs inversus, so complete reversal of all of the internal organs, just mirror image reversal. And that's not really associated with any problems necessarily from a medical standpoint. It's just that all of the organs are reversed. But this paper came out in 2007 that actually showed that a percent of patients with primary ciliary dyskinesia, rather than having normal situs or having situs inversus, actually had heterotaxy. So this was sort of the first description that there was this overlap between them. And some additional unpublished studies will indicate that about 10 to 20% of PCD patients have heterotaxy, so probably slightly higher than that initial estimate. And then if you look at the question of, well, how many heterotaxy patients then have PCD is really the cause of their heterotaxy, that's not known for certain, but again, some unpublished work estimates that probably 10%, maybe slightly higher than that. And as I said, that's important from a management standpoint because typically you don't think about sinusitis and ear infections and lung infections being a huge problem in kids with heterotaxy, but if PCD was what caused their heterotaxy, then those are going to be significant issues, and there's medical management that would be changed based on that. So the last thing, so those are kind of the things, when I see a newborn with heterotaxy, those are the things that go through my mind in terms of, well, what do I need to think about to try to give a diagnosis, to try to give the family some information about what the long term is going to mean for the child, and to help with the medical management if there's specific things. Obviously, again, if there's a genetic syndrome, there may be risk for intellectual development that isn't present if it's isolated heterotaxy. And then the last thing that's part of, I think, the clinical genetic evaluation of individuals with heterotaxy is giving the family some information about recurrence risk. As I said, the most common thing is that this is the first time that there's been anyone with heterotaxy in the family, and often parents will want to know, well, if I want to have more children, what's the likelihood that I'm going to have another child with heterotaxy? What's the likelihood that I'm going to have another child with any type of heart defect? And so we have empiric recurrence risks based, as I said, just on these epidemiologic studies. If you look at individuals with heterotaxy, how often is there someone else in the family? And that will tell us about 3 to 5% of the time. But really, the estimate is going to vary quite a bit, and we should be able to be more specific for individual families based on what the cause of heterotaxy is in that child. And so that requires us to think about all of those genetic things that I already talked to you about, because if it's a genetic syndrome, then the recurrence risk is going to be very different than if it's an isolated condition. So there is clinical genetic testing for heterotaxy now. As I said, that we recommend chromosome microarray testing be done in any newborn that has heterotaxy. And then I'm going to talk about ZIC3 as a gene that causes X-linked heterotaxy. I recommend that that newborns have testing for ZIC3. The main reason for that, so about 75% of families that have an X-linked inheritance pattern, meaning primarily boys that are affected, will have a mutation identified in this gene, ZIC3. And there has been a 4-gene panel that's available for testing, and about 20% of individuals will have something abnormal found on that. But the main reason for doing the ZIC3 testing is that it really significantly changes what I would tell a family about the recurrence risk. So if you do ZIC3 testing and it's abnormal or there's a mutation, then there's 50% recurrence risk in males, so very different than that 3 to 5% recurrence risk. But if you do it and it's negative, then you're back down to that 5% recurrence risk information. So in families who are wanting to have more children, I usually recommend this testing as a way to give better information about what their risk is going to be. So where are we in terms of just clinical utility of genetics for heterotaxy? Well, we're not where we want to be, but we're a lot better than we were 5 or 10 years ago. So we know now that if we do clinical microarray, about somewhere between 15% and 30% of newborns with heterotaxy will have something abnormal on microarray that explains the reason that they have heterotaxy. If we do the molecular sequencing, the proofreading type test, we can get somewhere between 10 and 20% that will of kids that will have something abnormal. So that leaves us, somewhere between 25 and 50%. I'd say probably closer to 25% would be how many children we can give a definitive cause to. And then the rest, as I said, is really on a research basis. We're trying to identify additional causes and trying to put the picture together to find disease associations. So the last part of the talk, I just want to go through, it's really sort of hardcore developmental biology and genetics, but for those of you that are interested in the research and interested in, well, how does this really happen? What are we doing to study this? I want to give you a little bit of background. So again, sort of the basic developmental biology, we know that there's at least sort of 3 flavors of how someone can get heterotaxy. You can get it because you have cilia abnormalities. We've talked about that a little bit. You can get it because of what we call cell polarity defects. I'll show a picture of what this means. And you can get it because of abnormal signaling when the baby is developing in the womb. So this is what these cilia look like if you look at them fluorescently. And there is, as the embryo is developing, there's this structure, it's called the node, or the organizer, and it's called the node because it does organize the left side and the right side of the body. It's there briefly in development before any of the organs form. It sort of sets up the pattern of the left side of the body being the left, the right side of the body being the right, and then it disappears and goes away. But it has cilia on it. And the cilia actually move. They rotate in a counterclockwise manner. And we don't know exactly how that triggers, but there's it causes a signal, a sort of like a chain reaction to occur, something about the beating of those cilia that helps the left side be established as the left and recognize itself as the left. So if those cilia aren't beating, or they're not beating normally, or they're not turning normally, then the left side doesn't get established properly. The cell polarity that I'm referring to, so those cilia, this is showing kind of a cell, and this is a cilia on a cell. So earlier in development, it starts out, it's sitting right on top of the cell, and then it needs to move backwards to be at a proper angle for the beating. And studies have shown that if it's not at the proper angle, then it doesn't make the fluid move the right way, and you don't get the left side developing the way it should. So, and then the last thing is, I said there's sort of a chain reaction that happens, and the technical term we use for that is signaling or signal transduction. And so that chain reaction that happens is one gene talking to another gene, and telling things to turn on. And we know a lot about the genes that are in this signaling pathway, and we know that many of these genes do cause heterotaxy. They cause them, we use animal models, and they cause them in mouse, for example, if we make changes in them. But when we look in patients, only a small number have actually mutations or defects in these genes. And again, my thought is that they're very important for early development. We're talking, the first 4 to 6 weeks of life when these, of embryonic life, when these genes turn on. And I think that many of them, if they're not working properly, probably result in a miscarriage. They're just too severe. So this is a picture, this is in a mouse, but this is what that node region looks like. It's sort of a teardrop-shaped structure. And we can look at signaling that goes on around it. And then this again is a mouse embryo, but what you're seeing here is this is the head, this is the tail. You're looking at it at the side, and it's kind of a U-shaped at that point in time. And what you're seeing is some staining that shows where a gene is being expressed. And this is the first time in a developing embryo that you can see any kind of gene expression where left side is different than right side. So this is only being expressed on the left side. And this has helped us a lot in terms of trying to study this, this signal pathway that occurs. So we know that all of these genes are important in this chain reaction. But what we don't know is when you have things being expressed on the left side, the heart is right here. How does that information get from the left side to the heart? We don't really know that, and that's one of the big areas of research. So talk to you a little bit about ZIC3, because it's a gene that we do know something about. It causes the X-linked form of heterotaxy, and this is sort of the structure, what the gene looks like. It's separated into different compartments. And we found mutations or changes in patients with heterotaxy. And so we made a mouse where the ZIC3 gene, we got rid of it. And what we see in the mouse is that the mouse has heterotaxy. So that's allowed us to try to understand during early development what's really going on, and why is this gene leading to heterotaxy, with the idea that if we understand what this gene is doing early on in development, we can find other genes that also are important for heterotaxy, and maybe we can figure out how could you intervene to change that. So this is a picture of the mice, and what we found after a lot of work is that this is that node that has a cilia on it, the teardrop-shaped structure, and when you're missing ZIC3 or ZIC3 doesn't work properly, that teardrop doesn't form correctly. So instead of having a teardrop, we've got sort of these three areas here of different cells. And so again, that the left side isn't going to be specified as left correctly because that teardrop-shaped structure hasn't formed normally. So the big challenge is, well, how do we get the teardrop-shaped structure to form normally? And that's kind of an area of focus right now. This is just another picture showing that normally this is sort of teardrop shaped. And when we look at our mice that are missing it, you don't really get that structure being formed properly at all. So mutations in ZIC3 cause about 75% of X-linked heterotaxy when you have that family history. If we look at just an individual who doesn't have a family history, about 3 to 5% of the time they'll have a mutation in ZIC3. And we typically think of X-linked conditions as only affecting males, but actually girls can have ZIC3 mutations too. So it's available clinically for testing, and you'll often see in families who have this, you may not see heterotaxy in someone else in the family, but you may say other birth defects, and it can be a pretty wide variety of birth defects, sort of GI defects, clubfoot, just isolated heart defects. And that can be confused with other types of genetic syndromes as well. So, and I just showed you the mouse studies that really show that the loss of function. So as I said, we're working to try to figure out how to correct that. So the last piece of research that we're working on is, as I said, we know the left-sided signaling, but we don't know how that signaling really gets transmitted to the heart to give it information. So when this chromosome microarray analysis first became available, before we knew that it was important to offer it in individuals with heterotaxy, we were looking at it on a research basis with the idea that we might find extra pages or missing pages that would really help us identify the genes that were causing heterotaxy. So we did a big study where we looked at 225 patients with heterotaxy, and we did this chromosome microarray on all of them on a research basis. And it was partly based on these findings that we decided, well, we need to be offering it clinically because there's enough individuals that really have clinically significant findings. But we also found a bunch of things that we didn't know how to interpret, and so we're using those on a research basis to try to see if we can identify new genes that are important for heterotaxy, and we've had some success with that. So we needed something that's faster to work with than mouse to do that, so we turned to frog. So we look in the frog and we see, well, is the gene expressed during development, is it expressed in the heart? And then we can actually inject frogs to knock a gene down, and then we can analyze them to see whether heart defects develop or not. And so as opposed to mouse, where it takes like 1 to 2 years to do this and probably $50,000, in frog we can do it in 5 days. And so the frog heart's not identical to human heart, but if we do, it's a faster way to validate some of these candidate genes, and since we have so many of them, we had 400 different regions that we're looking at. We're kind of going through using this, and if we find something that looks really promising, if we knock it down in frog and we see that it's causing heart defects or causing heterotaxy, then some of those will go on and try to make a mouse model of that. So I thought you might be interested in just seeing, so these are the frogs we use in the lab, and basically we just do in vitro fertilization. So when you gently squeeze a frog, they'll lay eggs. And then you mix it with sperm and they become fertilized. And this is probably 300 eggs or so that you're seeing there. And then we can inject them with a chemically modified structure that helps us knock down the gene that we're interested in knocking down. So that takes about a day and a half to do that, and then we watch them develop for 5 days, and then we score them to see whether they have heterotaxy or not. So this is what we use. We look at the heart. The frog heart has just a single ventricle instead of 2 ventricles, but we can tell by the positioning of it whether it's normal or not. Oops, sorry. And then we also look at the gut to see if the gut's coiled the right way, and then we look at where the gallbladder is positioned, if it's on the right side or the left side. And based on that, then we can say, well, this either affects just the heart, or it's affecting just the gut, or it's affecting all of them, in which case we would call it heterotaxy. So we have some interesting genes that we're following up on now. Some of them are related to cilia. Some of them are related to just early development, genes that we know are important in early development. And some of them are in signaling pathways that no one has really suspected as being important for heterotaxy before. So those will be interesting, I think, to follow up on. So those are sort of the research-based studies that we're doing. We are collecting samples from individuals who have heterotaxy and from their family members to do more of these genetic analyses to try to, as I said, understand additional genes that cause heterotaxy, and also for, as I said, the combination of genes that are going to predispose someone to heterotaxy. In order to get at that, we have to have large numbers, and unfortunately, heterotaxy is a rare disease, and so in order to get big enough numbers to begin to make these associations, which are statistical associations, we need to analyze a fairly large number of patients with heterotaxy. So we've done the 225. We have about another 100 that we have in process right now, and we're enrolling additional patients and families. We're also listed on the NIH, the clinical trials site as well. So if anyone is interested in participating, you can definitely feel free to contact me or talk to me after, but we'd be happy to include you in the study. And I'll stop there. These are the people in my lab who've done the work on heterotaxy specifically, and answer any questions that you guys have.
this is why you should go?
Yeah, I think that's a great question, and it's something that even between physicians we deal with, because I'm always trying to advocate to other physicians, like really genetics needs to see the baby, and I understand that it's not something that's going to impact the initial management. So I think I'm actually an advocate that if heterotaxy is diagnosed prenatally, which it is increasingly, that prenatal genetic testing be offered. Doesn't mean that the family has to do anything, but if with that information, but if it's a genetic syndrome that's causing it, so sometimes say trisomy 13 or trisomy 18 can have heterotaxy associated with it. That might impact the initial medical management in a critically ill newborn if you knew that your newborn had essentially a non-survivable genetic condition. I think with some of the PCD findings, there is going to be differences in medical management that would be recommended based on that. And I think once we understand that a little bit better, that will probably carry over to the intensive care side of things, because they want to know if there's something that's going to keep a child from getting off of a ventilator or cause them increased risk. So I think that's another thing. But the final thing is, I think that in most cases, it is sporadic heterotaxy. That's, if you had to bet money, that's going to be what the cause is. But most families like the reassurance that it's not a genetic syndrome, that it's not the intellectual development of your child is going to be as good as any other child, with the caveat that it's just going to depend on their cardiovascular malformation and the surgeries associated with that, and that can have some impact on neurodevelopmental outcome as well. But that there's not you're not going in with a increased risk for intellectual disability because of a genetic syndrome. And it's common enough, 25% of the cases have a genetic syndrome where that prognosis is going to be very different. So those are the types of things that I talk both with care providers and with families about. And we also know that if there is a genetic syndrome that's associated with or causing the heterotaxy, the sooner you begin to provide therapy that's directed toward that syndrome, the better the outcomes are going to be. So it would be much better to find out about that in the first year of life as opposed to at age 10. So, other questions? Yep.
So you had mentioned at some point you were talking about the celiac and how that they could cause heterotaxy. Now, obviously, PCD is there is overlap between heterotaxy and PCD, but even beyond that, are there other ciliopathies that are not necessarily PCD that may be causing heterotaxy symptoms?
Yeah, there are, and we don't really understand that connection very well, but the sort of complicated slides I showed with all the bubbles on them, and I talked about Bardet beetle, but any of those conditions can have heterotaxy as part of it, but it's a minority of cases, and we don't really understand that. It has to do, I think, with what the particular genetic defect is and what it's doing to the cilia. And we know, as I showed you from a developmental standpoint, that really it's the cilia at that node or organizer that are important for left-right. So if you get past that step of development, you're not going to have heterotaxy. So there may be, again, sort of a susceptibility issue where most of the individuals with those other ciliopathies somehow have enough that's working right that they can get past that step of development, but they're going to have problems with a number of other organs that have cilia that need to function later in life. All right. Well, thanks for your attention. Oh, one more.
So as you go in to see a geneticist, is it pretty much common knowledge what to test for heterotaxy, or should we as parents be prepared to say, this is what I want you to test for, or are they going to be, I'm just not sure if all groups are as knowledgeable?
Yeah, I think, I think geneticists get consulted kind of intermittently for heterotaxy. One thing about being a geneticist is that there isn't really everything that we see is rare. Everything we see is uncommon. So pretty much any patient that's coming to clinic, we have to read and prepare for, because and the testing changes so frequently that even if you just saw a patient with that last year, there might be new testing available this year. So they should come prepared, having read something or know something about it. I think it's always great when parents can advocate for what they know, and increasingly, because so much is available on the internet, increasingly I have families that come to me that say, well, we read this, this, and this, and we think, and hopefully you can provide some, as a geneticist, you can provide some clinical insight into, well, I think that's indicated, but this isn't. But I think they should at least have done, this stuff is all out there and published, and should have that knowledge. The piece that is under the radar a little bit, I think, is the PCD connection.